The overall goal of this procedure is to measure the function of a population of CFTR channels in a reconstituted system, and to examine the response of the channels to the presence of modulator molecules. This is accomplished by first purifying wild-type or mutant CFTR protein from SF9 cells using a rapid purification procedure, which yields functional CFTR protein in detergent solution. The second step is to reconstitute the purified protein into proteoliposomes of defined lipid as selected by the experimenter. Next, a chemical gradient of iodide is developed across the membrane by removing the extraliposomal iodide to generate a driving force for ion movement through the CFTR channel. The final step is to activate the channel and initiate iodide release from the proteoliposomes in the presence of the modulator molecule of interest. Ultimately, an iodide selective electrode is used to monitor CFTR channel function in real time in this reconstituted system.
This method enables study of the chloride channel activity of a purified population of CFTR molecules after reconstitution into phospholipid liposomes.
Although this method was designed to provide insight into the function of CFTR anion channel and the effect of small molecule modulators, this method can also be applied on other systems, such as functional characterization of other membrane proteins.
To be successful, individuals new to this method need to ensure that they have optimized the ratios of protein to lipid for their system, that they are not working with poorly functional or non-functional protein, and that they have generated a sufficient iodide